Biofilms are coatings of microbial slime, produced when bacteria settle on a surface and bind themselves together with a polymer goo. Biofilms shield bacteria from viruses and antibiotics that would otherwise kill them, and they crop up in many infections that plague humans. Outside the body, biofilms can infest water pipes, damaging industrial equipment. Timothy Liu, an assistant professor at MIT, was chosen for this year's TR35 because he has used the new tools of synthetic biology to destroy biofilms. He has engineered a type of virus called a phage so that it can feed on the bacteria in a biofilm, self-replicating and destroying the film layer by layer.
Bacteriophage are viruses, essentially, that only infect bacteria. They don't affect human cells at all. They were discovered about 100 years ago, and they've been shown essentially to be harmless towards humans. They're everywhere in our environment. And essentially they are either made out of DNA or RNA in terms of their genome. And they basically infect and either kill or live within bacterial cells. So many people have been trying to use natural phage for a long time to destroy biofilms or antibiotic-resistant bacteria. The problem with using natural phage on their own against something like a biofilm that has kind of a three-dimensional structure, I like to think about the biofilm as like a fruit jello, where the jello is the extracellular material that the biofilm makes to protect itself, and the fruit within are the bacterial cells. The problem with using natural phage or using antibiotics to try to destroy these biofilms is that they don't penetrate very well through the jello layer. It's very thick, prevents things from passing through it. And so natural phage gets stuck. They can only really infect the top surface. Antibiotics also have the similar problem. They can't diffuse all the way to the bottom and destroy the cells within the biofilm. So what we've done is to take a natural bacteriophage, look at its DNA genome, and find out where we can make modifications to it. We synthesize that DNA for those genetic modules, and then we basically paste it into the bacteriophage genome. And that gives you a basically synthetic virus that's able to infect the biofilm, replicate, and during replication, because it has a genetic module, it will produce a lot of these enzymes that can then chew up the biofilm, and therefore weaken the structure and allow basically more destruction.
Liu is also developing another approach in his campaign against biofilms, by making phages that enhance the effectiveness of existing antibiotics.
So I think you probably heard a lot about this in the news, things like MRSA or these superbugs that are essentially resistant to a large range of antibiotics that we're applying to them. The main issue there is that it's very difficult and expensive to find new antibiotic drugs that can kill these bacteria. So what we've done is take a completely different approach and try to use phage to enhance the ability of antibiotics to kill bacteria. And what happens is when you infect a cell with these engineered phage, they target those genetic networks that cause resistance. They knock them down. They suppress them. And therefore, when you apply an antibiotic on top of that, in combination therapy, you actually get much higher efficacy at killing the bacterial population. So we've found that you can kill up to 10,000, up to 100,000 times more bacteria than you would just using antibiotics alone.
The first commercial application of the phage technology could be in cleaning industrial equipment.
The original thought was to design these engineered phages for mostly clinical applications. So we wanted to treat biofilms that build up on in people's heart valves or medical devices, things like that. Or we wanted to treat antibiotic-resistant infections in human patients. Recently, we've turned our focus also to environmental contaminations caused by bacteria. So biofilms can stick to like metal pipes that we use in oil pipelines, that we use in HVAC systems and things like that. And what they do is they cause a buildup on the metal surfaces, and therefore a very poor heat transfer and basically loss of energy efficiency. So up to 80% of energy or the heat that you want to transfer through a pipe can be lost because of biofilms. So we're very interested in using the technology now to treat some of those environmental problems as well.
Liu has co-founded a startup to develop the technology.
So NovaPhage was started in 2008. It's basically co-founded by myself, James Collins at Boston University, and one of my former colleagues, Michael Karras. The focus originally was mainly on clinical applications. The company now has also developed a big interest in trying to use engineered phage against these environmental biofilm applications, because we think it has a lot of potential for improving energy efficiency in our homes, in offices, things like that. Currently, the company is quite small. It's a new startup company, but we have seed funding, so we're very excited to get out there and do some proof of concept models and get these products out into the market.